Hi everyone, here we are in the fourth lecture on microbial control, and you might remember that we're on chemical control, and so we're going to talk about a couple more chemicals that are relevant to this chapter. Here you can see we're going to be talking about hydrogen peroxide, and we've already talked about hydrogen peroxide, how it's toxic to cells, how it can damage our own cells, as well as bacterial cells. Any living cells can be sensitive to hydrogen peroxide, and it's because it oxidizes them and inactivates all sorts of chemicals that disrupt the metabolism of the cells. But hopefully you remember that cells also, or many cells, also contain an enzyme called catalase. And catalase takes hydrogen peroxide and converts it into oxygen and water, making the hydrogen peroxide inactivating the hydrogen peroxide. So when you use hydrogen peroxide for microbial control, you have to kind of flood the area to overwhelm any catalase that might be present. Hopefully you remember, let me just back up. Hopefully you remember doing the catalase test in lab where we could show that bacteria in the presence of hydrogen peroxide, if it bubbles from the oxygen, means it's making catalase. But if I just left this here and I kind of overwhelmed that catalase, those bacterial cells would die. And so it turns out hydrogen peroxide is very effective at microbial control. It's typically used as an antiseptic, and probably most of us have hydrogen peroxide in among our medicines that we would use for microbial control. Hydrogen peroxide is light sensitive, so you always see it in a dark bottle. And if you leave it in a clear bottle or can be exposed to light routinely, it will naturally break down into oxygen and water. But it's fairly stable. Your bottle of hydrogen peroxide will probably be effective for a year or more in that dark bottle. We have used hydrogen peroxide, humans, as an antiseptic on our skin for quite a long time, and it's very effective. It's one of my go-to chemicals if I have a small wound. You could use alcohol, but alcohol burns a little bit more than hydrogen peroxide. So a lot of times I would use hydrogen peroxide on shallow wounds. I remember my child having really chapped cheeks when he was a baby, and the doctor said, put hydrogen peroxide on it as an antimicrobial, and then cover it with something like a Vaseline kind of or Aquaphor kind of lubricant, so to keep it from drying out. But hydrogen peroxide is discouraged as an antiseptic in some situations because we've found over the years that it can slow wound healing, and that shouldn't be too surprising because when we're flooding an area with hydrogen peroxide, we're also damaging our own cells. So we can kill bacteria, but we can damage our own cells. So if you're treating a wound that's been difficult to heal, or it's a very deep wound where there's a lot of tissue damage, you might consider not using hydrogen peroxide because it can slow wound healing. If you work in a wound care clinic, you probably won't use a lot of hydrogen peroxide because you're getting people that are having problems with wound healing. So it's a good antiseptic to use around the house. Another thing that you might use hydrogen peroxide for is cleaning off countertops, particularly in this situation like where you're working in the food industry. I remember one time going to a cheese factory and seeing how they took samples of cheese out and they put them all over this counter and they test it for the number of bacteria. And then somehow you have to clean that up when you're all done. And hydrogen peroxide can be a really effective cleaner, and in that situation would be a disinfectant. And the reason is in the food industry, you don't want to get like any chemicals in your food products that you're processing. So to use something like hydrogen peroxide that can kill bacteria, but's naturally broken down in the presence of light into harmless things, oxygen and water, that's increases the safety of the chemicals in using the chemical in the food industry. So the question that we have in the lecture guide is, why does hydrogen peroxide kill more reliably on inanimate objects like tabletops than on wounds? And so it can be a very effective at killing microbes in both situations, but I would just say generally more reliably on inanimate objects. And think about that for a second and remind yourself that living tissue is going to have catalase. So you're kind of breaking down the hydrogen peroxide as you're using it, and inanimate objects won't have catalase. Okay. Oh, I had this article on chemicals that work against coronavirus, and you can look it up. There's probably some more recent ones than what I put right there. But I guess the one take home in that article was hydrogen peroxide can work. Bleach works really well. Um, let's see. The bleach is probably one of the best ones. And some people like to use kind of natural products, like use vinegar, because it's kind of less toxic. But it turns out vinegar isn't really great. We talked about how acids can be used to denature proteins, but it's not quite as effective against coronaviruses as things like hydrogen peroxide or bleach. Okay. Here's another chemical, and I would put this in the category of the most common chemical that you've never heard of in this lecture. And it says, describe how detergents, which we call, more specifically called quats, are used for microbial control. Um, you're thinking, what the heck is a quat? And a quat is a quaternary ammonium compound, a quaternary ammonium compound. And what that means, here we see this word ammonium. What a quaternary, meaning the fourth degree ammonium compound. Here's an ammonium ion. It's a nitrogen with four groups attached to it. So here's an actual quat that has this nitrogen with four things attached to a nitrogen. You know your product contains a quat when you look on the label. Now here's a more modern version of Lysol compared to some of those older ones that had phenol in them. But here we have this big long word right here. And I put it right here so you could see it. It says N-alkyl. Oh, it has kind of more on here than I wrote up here, but N-alkyl dimethylbenzyl ammonium. If it says ammonium, quaternary ammonium compound, it's considered a quat. And what a quat is it's a surfactant like soap. It breaks up oils on a surface so you can rinse off anything that's trapped in those oils. But unlike soap, it's also an antimicrobial. So soap, remember we said, will break up the oils, and so you can mechanically rinse off microbes off your skin. Whereas quats actually will break up the oils but kill the microbes in addition. And so this is what, when you have things that we call detergents, they usually have quats in them. Lots of your household products have quats in them. So go look at labels as your cleaning products, like kind of Mr. Clean. And I think even some, formulations of some of these pine cleaners, they're not using the pine anymore as the major way to kill microbes. They're adding quats. So a lot of these products that you use for, like these spray cleaners that we use around the house, they actually contain quats. They're very similar formulations. And so this one says, this is Lysol, and it says kills 99% of germs in 60 seconds. And it goes into a little detail about which ones if you look at the label and all that. So quats are really effective, and they're really common. They're a little bit too harsh to use on our skin, so we use them as disinfectants rather than antiseptics. They're probably the most common antimicrobials that we use in our homes because of their safety level and their effectiveness. The one thing about quats is that some microbes are resistant. And there's a genus of microorganisms we haven't talked too much about called Pseudomonas. And Pseudomonas is all over the place. This is something, it's in soil, it's in dust, it's something that we generally get exposed to. And Pseudomonas causes a lot of hospital infections. People with I just forgot the name of the disease. Anyway, people that have burns Cystic fibrosis. People that have cystic fibrosis that have, inherited a genetic form of this respiratory disease, a lot of times get Pseudomonas infections in their lungs, and they can be deadly. People die of that. Pseudomonas is a really common contaminant in, just anywhere. And so people that are burn patients can get Pseudomonas infections. When you have areas of your skin that are missing, Pseudomonas is one of the pathogens we worry about a lot. And so the the one of the precautions that's taken when somebody has like extensive burns and they're in the hospital, you're not allowed to bring flowers for them because so much bacteria can grow in that flower water. And Pseudomonas is one of them. So when they go in and clean the rooms or they clean out like the hot tub or the whirlpool where patients are being treated, using quats alone is typically not advised because people can get Pseudomonas infections and quats don't work very well against Pseudomonas. The next group of chemicals I wanted to talk about are heavy metals. And we're all kind of familiar with the concept of heavy metals. These pictures, or most of these pictures, are these pictures right here are all from your textbook. And I wanted to, just show you that heavy metals have been used a long time to help prevent microbial growth and prevent the spread of microorganisms. And so here it's saying under A here, it says copper in fixtures like this door handle can kill microbes for frequently touched surfaces. Eating utensils that contain silver can inhibit microbial growth. And there's even some incubators that are routinely used in laboratories that are lined with copper to prevent contamination of things in those incubators. This one says, let's see. Oh, I knocked off the last picture here. Okay. What did I put that? Why is that there? Anyway, one thing to know about heavy metals is that we're very sensitive. Living organisms are very sensitive to heavy metals. You've probably heard, things about lead in the water and all that. Well, lead is a great antimicrobial, but it's also a great anti-human. It can kill lots of organisms. And so one of the things we know about heavy metals is you only need them in really small amounts. And that can be seen in this visual right here. So here's a petri dish, and it's covered with, there's bacteria growing on it. All this cloudy stuff is bacteria. It's just a swab of bacteria and growing it up. And then what they did is they took, here's a copper coin and a silver coin and a silver charm, and they put them on the petri dish as the bacteria were growing. And if you look carefully, here they picked up this charm, and you can see underneath it, the bacteria did not grow. And here around this silver coin, there's a really thin area where you can see kind of a halo where the bacteria did not grow, and the same thing on this copper coin. What happens is when you incubate this overnight, a little bit of that silver or copper is going to leach into the agar, and it's enough to kill that bacteria. And I think that visual is really helpful to see how little you need to be antimicrobial. So a very, very small amount of silver can leach into this. I mean, how much silver leaches from a coin into an agar plate overnight? Not a whole lot, but it is antimicrobial. And as a result, silver in particular has been used a little bit more as an antimicrobial, especially as we get more antibiotic resistant bacteria. We're trying to find other ways to control microbial growth or control bacterial growth. And some of you might have heard of this cream called Silvadene cream, and it has silver in it, and it's commonly used to treat burns are a really common one, or wounds that kind of are similar to burns where you're removing like the top layer of skin. And it's because the silver is a great antimicrobial, but it also seems to soothe the tissues and allow for healing to occur. So it can kind of improve wound healing. So silver's been really effective. And here's another way you might see silver. Silver nitrate is actually this dark gray color. And you can imagine if this person has like a serious burn on their skin, you want to cover it up so you're not getting microbes in it. But the bandages themselves can be moist, they can be warm, there's dead tissue around. It's a great place for microbes to grow. So in some cases, they'll impregnate the bandages with silver nitrate and wrap the tissues in that, and that will prevent microbial growth. So heavy metals in very low concentrations can be effective for microbial control situations. Here, one of the chemicals I want to talk about are aldehydes. And you heard of aldehydes when you took your intro chemistry class, and aldehydes were one of those functional groups. Now, you don't have to memorize this again, but it might look familiar to those of you that have good memories. There's a carbon with a double bond O and an H, so an organic molecule with that on it is called an aldehyde. And aldehydes, it turns out that this chemical group is really unstable, that it just kind of gets disrupted really easily. And you might remember when you disrupt chemical bonds that you get unpaired electrons, and that means these groups are going to want to bond to other things. So they can disrupt proteins, they can disrupt disrupt nucleic acids in cells. So aldehydes are very good at killing living organisms. They can be bactericidal, virucidal, kill viruses, fungicidal. So aldehydes can be really effective. Here it says for aldehydes, long-term exposure, which is about 10 hours, can kill endospores. So enough exposure, you can sterilize. So aldehydes can be used for that. The aldehydes that you've probably heard about, probably when you think of aldehyde, you think of formaldehyde. And we use formaldehyde to preserve cadavers, whether they're humans or animals that we're going to dissect in a laboratory situation. And so formaldehyde, it kind of firms up the tissue of the organism, and it kills microbes. So you can get that, fetal pig or human cadaver if you had gross anatomy for weeks and weeks, because without it decaying or rotting, because you can kill all the microbes using formaldehyde. Now it turns out formaldehyde, and we said aldehydes are pretty toxic. They interact with all sorts of molecules. So formaldehyde is really dangerous. Here's somebody this is actually a Civil War photograph of somebody using formaldehyde to embalm a body. And that was one way that they returned soldiers back to their homes or deceased soldiers back to their homes to be buried. And so here's a guy kind of putting some formaldehyde into this person who had died. Even like I said, the animals that you might dissect in the laboratory have been treated with formaldehyde, but formaldehyde we found is just too toxic for us to be using, in a teaching lab like that. So what you do is the company that you buy them from will take the animals and then soak them in all sorts of other alcohols. And so the formaldehyde did kind of the initial complete sterilization of the animal and the firming up the tissue, and then the alcohols will help soak out the formaldehyde. So when you get, and still be somewhat antimicrobial, but they just evaporate more easily. So when you get like a fetal pig or a sheep brain to dissect in A&P lab, most of the formaldehyde is gone. There's another aldehyde that's common, it's glutaraldehyde, and it's not quite as toxic as formaldehyde, but it's still also too toxic to be a home-based product. And so glutaraldehyde, the one place you might find that in a hospital setting is in some of the cleaners. So this stuff called Sidex, if you looked at the active ingredient, it would say glutaraldehyde. And here it says sterilizing and disinfecting solution. So sterilizing, that's a big deal. So if you have a patient that has C. diff because they make endospores, that would be a product that you could use to clean the room. The other one, if you remember from our chart of what organisms have the most, are the hardest to kill, Mycobacterium, and particularly Mycobacterium tuberculosis was up there. And we said that's hard, those cells are hard to kill because they have a waxy wall. Sidex will work against that. So these can be used. They're not typically used in the home setting because of their danger, but because of safety issues. But they are routinely used in microbial control in certain situations. This is my last chemical I wanted to talk about, and it's called ethylene oxide. And ethylene oxide is actually a gas, and it's very effective at microbial control. And here it says this gas called ethylene oxide, it oxidizes, kind of like hydrogen peroxide does. And it sterilizes, it can sterilize things in 4 to 18 hours. Now, here we go. It's one of the most common sterilization methods in the healthcare industry. So again, something you might not have heard about, but it actually is quite common. And when you think about it, the advantage of having a gas is if you have a heat sensitive item, a gas can kind of go around into the nooks and crannies without damaging the item with heat. If you have something that's got a lot of nooks and crannies, like the Apollo spacecrafts that they sent up to the moon and then they came back, at those times we didn't know if microorganisms existed in space or on the moon. And there were a lot of issues concerned about how do we decontaminate things to make sure that they're not going to bring back some pathogens and wipe out humanity. These were real concerns. And so here you can see like this Apollo space capsule has a lot of little nooks, a lot of little spaces. So if you can pump a gas into that to decontaminate. In the healthcare industry, a lot of those paper products that you use are sterilized, you know, the gowns and things like that can be sterilized using ethylene oxide. You might have electronic tools that need to be sterilized, maybe tools used in surgery. They can be sterilized using ethylene oxide, this gas. And that's kind of handy, you know, if you can sterilize and not damage the items. But one of the things to be aware of is that ethylene oxide is a dangerous gas. It can cause cancer, and it is explosive. So you have to use it under very highly controlled situations. So this chamber here looks kind of like my autoclave. And it is the same kind of stainless steel chamber. And you put all this stuff in there, you close the door, seal it off, and then the ethylene oxide gets pumped inside of it. It sits in there for a certain amount of time, and then it's pumped back out in a very controlled manner. So this would not be something you would do at home, but in a hospital setting or an industrial setting of some kind, it could be very important. Do you remember the other chemical we had that could be used as a gas? Do you remember chlorine? We use chlorine gas. And I believe, oh, there's a matching. Chemical methods of control matching. I would recommend you try that, and I think if you stop right now, you can do that before I give you the answers, which I will do right now. There we go. There they are. I'm pretty sure those are the right answers. If you have any debates about that, bring it to class and we'll talk about it. That is the end of microbial control. So I'm not going to go over that one. But that's the end of microbial control. And I'll see you soon and we'll talk about it. Take care.